From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on Halloween, October 31st, 2025. Hello, Micronauts and trick-or-treaters, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 115th episode of Matters Microbial. Thank you for every view or listen, and please spread the good microbial word. My favorite holiday is Halloween, as you might guess. Here is a microbial mask that my wife, Dr. Jennifer Quinn, made for me using giant microbe plush toys. I guess it's true that I'm a microbe biologist. As I mentioned last week, I recently gave an exam to my micronauts in my microbiology course here at the University of Puget Sound. On the exam, I asked them what most interested or amazed them thus far in our course, and I was not disappointed by their choices. As you can see here, one micronaut was amazed at the coexistence and coevolution between animals and microbes. A second micronaut, seen here, was truly interested in kefir grains composed of yeasts and bacteria, and how that delicious obligate association evolved and what it could mean about the nature of multicellular organisms. And finally, a third micronaut seen here was amazed by the fact that enormous bacteria can have tens of thousands of copies of its genome within the cytoplasm, and how those copies maintain their own fidelity during replication. I'll put links in the show notes of the readings that I gave my micronauts that spurred these responses. Again, finding out what excited or interested students truly matters to me as an educator. As I've said repeatedly, I've enjoyed teaching micronauts every year, and seen here is a bit of Lux art created by one such micronaut celebrating their new microbially centric viewpoint, Micronauts Forever. Indeed. And finally, one of my micronauts is quite artistically minded. Here you can see a bit of art he created during class, showing the enormous bacterium Thiomargarita magnifica superimposed over a diagram of a genomic island. Jonah is very skilled and creative. Thank you, Jonah. One of my interests for many years, research-wise, remains predatory bacteria. That is to say, bacteria that pursue and consume other bacteria. Since I am at an undergraduate institution, it's been a little challenging for me to accomplish much research in this area, but I continue to try and the students love it. A tip of the microbial hat to prior Matters Microbial guest Dr. Laura Williams, who was on the podcast last year to discuss the work she did with undergraduate students on the bacterial predator Delavibrio, an old microbial friend of mine, and a topic for today's podcast. Delavibrio is a tiny, fast-moving bacterium that attacks other gram-negative bacteria, invading their periplasm and growing at the expense of their prey. Serious, folks, you have to understand that when Delavibrio hits a prey cell that you're watching under the microscope, it knocks it out of field and you have to go find it. Here's a photograph that I helped to take many years ago, tip of the hat to Tom Pitta, at the Microbial Diversity course at the Marine Biological Laboratories in Woods Hole, Massachusetts. It's a feeding frenzy, and you can see the Delavibrio cells on the attack here. I feel so sorry for the prey cells with many Delavibrio cells attached to them. And here's a close-up of Delavibrio attacking E. coli and invading it, taken at the same time so long ago. The concept of this tiny predator is very intriguing to me and to students, and I think to the general public. And here's a claymation video my wife, Dr. Jennifer Quinn, made quite a while ago, while the theme from the movie Jaws should be in the background playing. You can see the invasion. You can see the elongation of the invading cell. You can see it septate, grow flagella, burst the cell, and come storming out, including one that smiles at the camera. It's true. My wife is very talented. Which leads us to today's podcast guest. I am beyond excited to welcome Dr. Hannah Ledvina, Assistant Professor at the University of Michigan, to the podcast to discuss her research findings on how bacteria can protect themselves against attack from several types of predators using a type of a shield. Welcome to the Quality Quorum today, Hannah. I am so excited to chat with you today.
Thank you so much. I'm very excited to be here and chatting about predatory bacteria, my favorite topic.
Well, the first thing that I would like, and your agreement on, I shouldn't automatically say I want your agreement on, is it turns out that when predatory bacteria were first discovered, they were thought to kind of be exceptions to the rule. But just like in nature, there are many approaches, and Delovibrio is simply one. I know you have an interest in the myxobacteria. There's the elusive Vampirovibrio and Vampirococcus, which I don't think anyone's ever cultured.
Nope, I don't think so, yeah.
Have you heard of anyone culturing it? And then I think there's one that's the holy grail of predatory bacteria. I don't know if you're familiar with it, Daptobacter.
Yes, that is one that I would
just rams.
Love to figure out. Amazing.
Rich Guerrero in Spain claims that he has a culture of it, but when I've asked for samples, this is many years ago, he never replied. So there you go.
Bummer. We'll just have to go start digging in the dirt ourselves.
Yes, it's exactly what Laura did, and I think it's a really good idea because they're literally everywhere. For listeners and viewers, the way that you look for predatory bacteria, typically, at least the types that I've been familiar with, is you treat it as if you're looking for bacteriophages. Now, bacteriophage plaques, as they're called, reach a certain size and stop when the cells cease growing. But predatory bacteria continue to nom. So those plaques get larger and larger. And that's how this was first discovered in the 1960s.
Absolutely.
So I want to say congratulations on your new position. How does it feel to be an assistant professor?
Oh, it's absolutely amazing. You feel like you finally made it after decades of trying to get there. It is an overwhelming process, but I'm having a lot of fun trying to get everything up and running, starting to hire my first people, and it's a very exciting time for sure.
Now, I am going to be putting up links to your faculty site and your laboratory site as well. And I really encourage listeners and viewers to take a look because Hannah doesn't just work on things like Delovibrio. She has other interests as well. So I think you have such a wonderful program, and I am applauding you, Microbial. These are my flagella clapping for you.
Your flagellas are happy. I love it. Thank you. Yes. I love all things. Microbial warfare is what I like to think I study. The microbial world is hardcore, and we're just really starting to understand how it all works.
So you know that expression red in tooth and claw about nature? Nature's red in tooth. We need to have the equivalent. It's red in pilus and type 6 secretory systems. We need to get more elegant than that.
But it could be there. I like it. We'll work on that.
So there's an expression I keep saying during the podcast. I don't know where it's from, and the expression is a thumbnail sketch. Can you give us a thumbnail sketch of your research program?
Oh, absolutely. So I study what we think of as the bacterial immune system. So, we are constantly bombarded with different pathogens trying to make us sick. And to counter that, we've evolved these incredible, intricate systems that specifically detect and defend us against these pathogens. Turns out bacteria also have these amazing immune systems that we're only really starting to appreciate. Bacteria out in nature are constantly being faced with their own viruses. These are the phages we've already talked about a little bit, as well as these things called predatory bacteria. And so I'm really interested in just trying to understand how bacteria protects itself from all of these. And there's been this really fun overlap where we're starting to see that a lot of the ways that bacteria protect themselves are actually the same as how humans are doing it. And so we kind of see this really interesting evolutionary line of how immune systems evolved and what is kind of like the best way to defend yourself. And so we're really interested in figuring out what makes up the bacterial immune system, and then how do all of those systems come together and work to keep a bacteria alive.
You've made me very happy already in this interview, and I'm going to tell you why. I've been called a microbial centrist, and I am. And one time I was critiqued that I was only these are botanists telling me that I was only interested in microbial things. I said, no, plants do plenty of interesting things. It's just most of those really cool things they stole from bacteria and archaea, which is 100% true.
So true.
And when we start looking at many of the mechanisms by which bacteria and archaea navigate their microenvironments, we see echoes of it in ourselves, as you just mentioned. And I think it is beyond fascinating to remember that.
Absolutely. I feel like bacteria have it all figured out, right? They've been doing this for billions of years. Why do we pretend that we were the ones who came up with anything, really? They've had it figured out for so long.
So feel free to use my motto, first evolved, last extinct, because that's what it is.
Ooh, I like that a lot. I'm going to use that in the lab for sure.
I will send you a button that has that on it. Don't you worry.
Excellent. Can't wait.
So I love this idea. Now, I will tell listeners and viewers that what used to happen to me a lot when I would talk about Delavibrio at scientific meetings is people would think of it as a superbacteriophage. The difference is, of course, Delavibrio is actually using ATP. And as of the time of this podcast, I know of no virus that is actively generating and using ATP. Fair enough?
Yep.
It may change next week.
Oh, always. Yeah.
Right. So, and then they would treat this as if it's a superbacteriophage. So they would say, well, let's find out what's different and important and how bacteria can protect themselves. I remember this very well. There's a quite nice scientist named Susan Koval who did a series of papers in the 1980s and 1990s about this part of the outer envelope of E. coli is not required for predation.
Mm-hmm.
So things like S layers, I was always taught that S layers were part of it. Turns out it doesn't matter.
Mm-hmm.
LPS doesn't matter. All these different things. And so it was really a mystery of how Delavibrio did its business. There's been some recent work, and I will put a link in the show notes to people who think they have figured out how it is that Delavibrio recognizes its prey, because Delavibrio is able to recognize a very broad series of potential prey organisms. They pretty much only are interested in gram-negatives that have that outer and inner membrane, and the region between is where they grow. Certainly that's true for Bacteriovorus. So with that in mind, people would say, just look for an E. coli that's resistant to attack, and then you'll know what the gene is. And, you know, that occurred to a lot of people, including yours truly, and it didn't work very well. And that's what really excited me about your work, because you did find something that took you there.
Yes, it was a long journey, but we did get there eventually, yes.
And I think that the key to that is twofold. The first is to recall that Delavibrio's, quote, natural prey is not a lab rat. That is to say, if you've cultivated E. coli in the lab for decades and decades and decades, it doesn't necessarily reflect what you would see with a freshly isolated, I'm going to say cultivar, but that's not the word. You know what I mean.
Exactly, yes.
So please explain what you did, because I love the diagram.
Wonderful, yeah. So our approach was really to kind of learn or take from what we have learned from the phage defense field, and that is that most bacteria don't carry a full set of their immune systems at all time. It's more a large community comes together and they work and each have a little bit of the immune system, and as a community they can protect themselves. And so then our idea was, well, we just really need to start surveying the diversity within E. coli to try to find that rare instance where Delo defense is now actually present in the genome. And so we were able to get access to this incredible collection that has been around for decades now, and people have kind of used to characterize different aspects of E. coli. It's called the E. coli Reference Collection. It's a set of 72 strains isolated from all around the world, all from natural sources. Importantly, they're all non-pathogenic, making it very easy to work with them in the lab, which is always a helpful aspect of this. And we just started looking to see if any of these had defense against Delo. And starting out, this felt like such a fishing expedition. We fully did not expect to get anything. We were like, this is a crapshoot. Like, you never know, though, we might find something. And so then we were fully shocked when it started to be that roughly a third of the strains we would look at had insanely potent defense against Dello. I could not get Dello to infect these E. coli no matter what I did, and I tried very hard. I would do all sorts of things to kind of force this interaction to happen, and we could just not get these E. coli to die from Dello. And so I think this is exactly what Mark was talking about, this idea that, most of the Dello work has been done with these strains of E. coli we just keep cultivating in the lab. And we know from genetics that over time, they're going to start kicking out the genes they don't need. And so if you don't threaten them with whatever those genes defend against, they will lose them. And so just going back to these natural sources where they were more likely to have encountered these predators more recently, I think we were able to stumble upon these strains that had continued to have this regulation present, and we were able to start uncovering defense. And so, we were able to find a bunch of E. coli strains that were resistant, and it was really exciting. And so then what we tried to do is figure out, what are the genes responsible for this? And I'll remind you, our hypothesis really was going to be that this is like this rare gene that's only present in some E. coli, and we were just kind of lucky to stumble across it. 100% not what we found. Instead, as you can see in this diagram, what we did is we took one of these resistant strains called ECOR14, and we performed this technique called transposon mutagenesis, where basically you can go through and randomly disrupt all of the genes throughout that strain. We then created a gridded library, so each well was an individual mutant of E. coli, and then we individually infect each of those mutants with Dello to now see if they've lost the ability to protect themselves. And so we screened thousands of these mutants. As you can imagine, this was a lot of work. This is where one of the reasons I love Dello comes into play so much. I had so many undergrads able to come into the lab and gain experience by doing these assays. They were able to learn so much, and I was able to get thousands of mutants screened, quite rapidly. But from all of these different mutants that we screened, we ended up only identifying 15 strains that had now lost the ability to protect themselves. We can do sequencing techniques to now map to figure out exactly what genes have been disrupted and start trying to piece back together what defense was. This was the hardest part of this project, was trying to link back all of these diverse hits to one central pathway or component and understand what was going on. So again, we really thought maybe we would just hit this brand new defense system, and so all of the mutants would be in that. Instead, we were hitting all of these kind of core regulatory factors that E. coli have. But when we mapped them all up, they all centered on the production of this absolutely incredible structure called curli. And so I have a diagram of this you can see here. Curli are these, what we call functional amyloids that bacteria can make and put on their cell surface. And these amyloids have most famously been implicated in playing a role in biofilm formation, which I know you guys like to talk about a lot. So this is where bacteria come together in a community. Curli fibers are thought to help kind of structure that that matrix so that all the bacteria can stick together. And so we were finding this, this structure that was already kind of well understood, and we thought we knew what it did, but now it seems to be playing a role in defense. And so this was really unexpected for us and kind of took us down a new avenue that we weren't expecting. But so we were trying to figure out, so we're making these random amyloids, we're defending against Dello, exactly how is this all working? And so we turned to, just kind of looking at the system through microscopy to see if we could start figuring out what was going on. And so we used something called transmission electron microscopy. There's an image here that I'll show you that it's going to be color-coded to help us kind of like break down exactly what we're looking at in each of these images. But basically what we found is when we looked at these resistant strains being infected with Dello, we saw the E. coli, which are shown here in green, and then we have our little Dello that come in and infect. These are in orange. And then what we found is curli here in purple just completely encases these E. coli in this suit of armor is what we call it, this molecular suit of armor that is encasing the E. coli and blocking the ability of these predators to come in and even start their infectious cycle. Here's another image where now you can see if we get rid of that curli, the dello is kind of free and or not the dello, excuse me. The E. coli, you can now see here that the E. coli is free and its membrane is exposed, and dello is able to come in that really intimate contact with its outer membrane and start that infection cycle. And so we really just, this was completely unexpected. We're finding that this part of E. coli that's kind of inherent to E. coli's biology can be used as a shield to now protect it from all of these different pathogens that E. coli has to face out in nature.
So this is very interesting for a number of reasons. First off, everyone should know that Dr. Ledvina's original title for this is Suit Up, which is just perfect. Okay? I love it so much. Secondly, I want to remind listeners and viewers that if you were to take E. coli and you would take bacteriophage lambda, because I've done this quite a bit, and you want to look for E. coli that's resistant to this particular type of bacteriophage lambda, mutationally, if you alter its receptor, which is the maltose receptor in E. coli, LAMB, what happens is that now the bacteriophage can't get in. Now, this is the paradigm that everybody kind of applied to this problem. There has to be something specific that's being recognized. And what I like about this work is it's a general defense response, because the curli doesn't necessarily interfere with solutes passing in or out of the cell membrane. So as you would get overheated in a suit of armor, if your armor is too tight, as it were, well, heck, look at the mycobacteria. That they have mycolic acid, which is super hydrophobic, and it really slows down their growth rate. That's the trade-off. Makes them all slippery to your macrophages. So in this particular case, you found that these, I'm going to call them undomesticated. I don't know if that's fair. Undomesticated.
Yeah.
Do you?
I've used that term. Yeah, I love that.
I have been saying that I love undomesticated bacteria my entire career, And that's because, and there's nothing wrong with using a well-defined system. I don't want the coli heads to come hit me over the head, but it's true. You can learn new things. Now, what's interesting here to me is that Delovibrio, at least strain 100, which you're using, is that correct?
Exactly. Yes.
And strain 109 that I cut my teeth on Anyway, that I learned to use many, many years ago. They were originally isolated not on E. coli, but on Pseudomonas. And I have thoughts I'll share with you off camera about that. But my question is, do you have the curli situation taking place in other bacteria? Have you looked for it?
We have started looking at that. That was a big part of this publication was, trying to understand what bacteria are now making these amyloid proteins, these curli-like proteins, who's making them, and are they all being used as kind of this defensive suit, or can they play other roles? And so we found, teaming up with Arvin at the NCBI, so many bacteria make amyloids, way more, of course, than we ever appreciated, because genomics is just continuing to show us we have kind of underestimated bacteria, I would say. And so there's many more species that we realized are making them, and there's kind of these different flavors of curli now that we can study. And so Pseudomonas actually has its whole own flavor of amyloids that it makes. And so we're just really starting to play around with those and try to understand exactly what's going on in those different organisms. But that is something we are really excited to dive into and better understand.
I think the folks who isolated these strains originally, Schatz and other people, were using Pseudomonas putida. And I have many thoughts on Delovibrio's life cycle between those two things that I can share with you later. But it's very old school micro there. So in any event, you're using a word that I know some people are going to trigger on, which is the term amyloid, which they've heard in another context in the news.
Yes, definitely. So these are highly related to the amyloids you have probably heard about. So we typically, as humans, associate amyloids with the fact that they can form plaques in our brains and cause lots of neurodegenerative diseases, Alzheimer's, things like that. These are when human proteins misfold and cause amyloid plaques in our brains. Turns out bacteria make these exact same types of protein folds, but they make them on purpose. So we call them functional amyloids because we kind of think of the ones in humans as like accidental amyloids. You don't want those, they tend to do bad things. But bacteria and certain archaea and fungi, I believe, also make these amyloid-like proteins. But now they have a purpose and a role in biology that we're really just starting to try to understand a little bit better. Like I said, historically, it's mostly thought to be an adhesive role in
Mhm.
biofilms. Amyloid proteins are sticky, they bind to each other. But I think we're also starting to better understand that it's kind of a much more diverse biological tool that, of course, bacteria have played around with and kind of evolved interesting ways to exploit this kind of challenging biochemical protein.
If you look back to the Koval paper, the paradigm is that the S layers on the outside of many gram negatives could act as a generalized protector. And people have talked about that to kind of fend off protozoal predation on bacteria, that kind of thing. But remember that in the process of cultivating these bacteria from the environment, they often lose S layers. So it might be you're coming to the same conclusion from different directions. And that being that there can be a generalized defense, but there's a cost to that defense. So when you raise things in the lab, they can dispense with that phenotype, as it were. And I do suspect a lot of that goes on. Because if it didn't, then you would have everything would learn how to do that learn, you know what I mean, evolve
Yeah.
or share to do that. And then there wouldn't be any bacterial predation anymore. So it's the Red Queen all over again.
Yep, absolutely. We're really trying to figure out what the exact cost here of making the Curli amyloids is. Amazingly, we don't see just like an immediate fitness cost in the lab, but as you can imagine, something like motility is probably going to be severely impaired if you've wrapped yourself up in sticky proteins.
Yeah.
And so, absolutely, it's this trade off where in the lab, maybe it's more beneficial to be able to swim. And if you don't have Dello around, why keep Curli, which is also going to cost a lot of energy to make and produce? So, you would just start turning it off. And so, I think, again, you just kind of have to we were able to game the system by screening so many different E. coli that had been coming from these unique environments that allowed us to find that right combination of right environmental factors where we were able to see this.
You know, historically, when people were studying Delovibrio, gosh, I'm old. What they would do is they would use a particular type of E. coli. In my case, it was ML35 that had been propagated in the lab for I couldn't tell you how long, or MC4100, which is more defined, but again is a lab rat of
Mhm.
and I like rats, so I'm not being negative about this. Um, of an organism, and you don't get an insight in how things look. This is something I teach my class a lot, that domesticated bacteria are quite different than their original isolates, and there's like a wealth of work on that from Losick and Coulter in particular at Harvard, and it's really solid work. So we tend to think one-on-one predator prey, but that's not really how it works. And it's
Yeah.
important to remember that.
Yeah, absolutely. And kind of adding on that, what has been pretty cool is we can actually kind of reverse what the lab conditions have done because we can now take our lab rat strains, tell them to turn back on their curli, and they become much more resistant to Dello. So it really is, they're all ready to do it as long as that evolutionary pressure is there for it to be appropriate for them. Bacteria are clever.
It is. And I want to say that historically, when you said, and I haven't looked to your techniques, but the way that we simply did this in shake flasks, you would get a cleared culture. You could
Yep.
always recover prey organisms from that, even though it has destroyed most of what's there. And I often wondered because then you could retest those and they're no longer resistant. That was one of the first things that everybody did. So there may be a lot of what I'm going to call variable gene expression going on all the time.
Absolutely, and that's really what we kind of came down to with Curli is there seems to be we kind of call it this bet hedging strategy that E. coli
Yeah.
are playing. Every single population of any strain of E. coli that we have looked at least some cells are making a lot of Curli. And thus, they're going to be able to protect themselves from that invading population, be that small population left at the end of your culture. But then there seems to be something special about some strains of E. coli where they're 99% of them are making a ton of curli. And that's how we were able to really kind of piece apart what is the defensive mechanism here. But it's true that every single E. coli or any bacteria that in theory makes curli has that potential to protect themselves and probably can kind of squeeze through a few last cells.
It's so interesting to me, Hannah, because Eduard Djurkovic in Israel had written a paper about what he called phenotypic plasticity, because what he's dealing with is the fact that you can recover E. coli from these cultures. And this is a little different than what you see in a phage thing. And he was trying to figure out what was going on. And I think you did.
We have met with many of the people who have kind of attempted to, and I think we're all kind of honing in that a lot of the things might have been because even our lab strains of E. coli are making a little bit of curli, and that is just enough to kind of keep everything around. Because then if you completely knock out curli from those strains, you don't really see that tiny little population sticking around. And so
I agree. There's also a philosophical or a perceptual issue because we tend to be reductionists as scientists. We shouldn't be, but we are. And I'm not putting that down. Sometimes you get to answers that way. And you think, for example, and this is one of the things that bedevils my poor students, is that every member of a bacterial colony is not precisely the same. And there's variation that takes place all the time. Look, historically, students see it when they isolate bacteria from nature and the phenotypes aren't stable sometimes. And there are all kinds of explanations for it, and it all comes down to domestication. And that's because the originators of all of this, and I'm not being negative about it, wanted stable phenotypes that didn't change, but that's not what nature favors.
Exactly. Yes.
Bet hedging. I love how you put that.
Yes. They're thinking ahead. They're smarter than we are, for sure.
Well, they're certainly older. I get accused all the time of anthropomorphizing microbes, and I'll say yes.
Yeah, I get called out all the time, and I will never be able to stop. It makes my job way more fun.
I agree completely. It's so exciting to think about these things. And when I talk about what a bacterium perceives, people get really unhappy with me there, but they shouldn't be, because they perceive differently than we do. Their whole biophysics is different.
Yeah.
So there's a bacterial predator that I try and study called ensifer, and it does this thing called tracking, where you put down the prey organism and it follows wherever you put the prey organism. So students do their initials and it gets all gooey. It's really fun. But it's cute. You turn the plate this way and it goes up and you think, oh, they're like mountaineers, microbial mountaineers. But they're not from the point of view of the very tiny. You know what I mean? Yeah.
Yeah.
Right. And we don't think that way because we're large. We're macrobes. Yeah. And that's what it comes down to, I think. But I love this work so much because this isn't that was my biggest fear, is there would be something specific going on, and it couldn't be because of the host range of Delovibrio.
Absolutely.
But I was so excited by this work when I saw it, and you were to be so applauded and congratulated for this wonderful, wonderful paper that we're certainly linking to. I started to say leaking, but no, it's already published.
We'll leak it too.
So one of the things that I want to reemphasize is there are lots of strategies for predation, and I know you've tried to look at this particular concept with other types of predators, and perhaps you could say something about that.
Absolutely, yes. So we're interested in all the different types of threats that bacteria are facing, and that is a whole diversity of bacterial predators. You know, just like with humans, we have like our extracellular bacterial pathogens that bind to our cells, and then we have our intracellular bacterial pathogens that invade into us and can infect us from the inside. Bacteria have the exact same thing. We've been talking about Delo, which I think of as an intracellular pathogen. It gets inside the bacteria to grow. But bacteria also have extracellular predators. We've talked a little bit about these vampire bacteria that come in and bind, and they literally will drain the nutrients out of their host. Just like a vampire, they're the coolest things.
Yeah.
But we wanted to start exploring the idea of extracellular pathogens of bacteria, especially because we saw Curli acting as this physical shield. We thought, okay, maybe this could then defend against anything that kind of needs to come at you from the outside. And so we turned to Myxococcus xanthus, which is just another one of these organisms that you love so much if you love bacteria. They're just so fascinating. These ones perform something called wolf pack predation, where they come together as a swarm of bacteria and work together, and they'll eat up an entire colony of E. coli kind of working together. They like swallow it, and it kind of looks like a wolf coming and eating it. It's absolutely amazing. And Myxo is another one like Dello that is thought to be this generalist predator. It can eat all sorts of different bacteria. And so we wondered, can Curli do anything to block this? And indeed, we found that Curli serve as a very potent barrier to Myxococcus as well. So Myxo, as you can see from this diagram, has many different ways that it predates on things like E. coli. It secretes a bunch of things, as well as has to come into direct contact with those E. coli cells to start injecting them with different toxins. And we found that Curli can block a lot of these things. And so our strains that make a ton of Curli are significantly less susceptible to Myxococcus than strains that lack the ability to make Curli, or some of these more domesticated E. coli that we work with in the lab. And so I love to think of Curli as like it's your first line of defense for bacteria. It's maybe like our skin. It's just the first thing that a pathogen has to overcome and kind of penetrate through in order to really start establishing that infection.
So this is very interesting to me, because I love my Myxo. Dale Kaiser taught me a lot about Myxo all these years ago. Wasn't he the nicest human being in the world?
Yeah, you can't find a nicer person. Yeah.
There's a hysterical thing where someone did like a fake rap video about glycolysis with him. Have you seen it?
No, I'll have
And
to look that up.
and he looks so completely confused. Why are they doing this? And he's such a sweet man.
Yeah.
I really wanted to do a postdoc with him, and he was so nice I could have browbeat him into it, but no, you don't do that, right? You treat him nicely, because there are so few people. What I'm getting at is that I'm wondering what the cost of the Curli are to the bacteria. And I'm not doing an all or nothing type of argument that there's a cost to everything and you only make it if you need it, because that's inferring intentionality to evolution, which I know I'll get in a lot of trouble for. So what I will say is, does it affect high molecular weight potential nutrients that come into a cell? So I'm thinking about oligosaccharides.
Yep.
And I know that E. coli can use oligosaccharides. So I'm wondering if Curli could interfere with that process. And that might be the fitness cost that it would have.
Absolutely. We have not, I would say we're really just starting to push at what could be the cost, and we have not tested that. So that would be really interesting. What we do know is that for the most part, Curli doesn't seem to block phage infection. So phage still seem to be able to wiggle their way through and find their receptors pretty much just fine.
Yeah.
So we're trying to understand if it's like a true size thing. And so we're starting to do some live cell imaging to really understand what happens to that Dello when it encounters a cell. Does it get stuck? And maybe that's more what's happening. Does it bounce away? Really try to better understand the dynamic when they come into contact, to then maybe start to better understand like, how can this play a role as a barrier, and how selective of a barrier is it?
It's really interesting you say this, because you can literally watch a Dello, and I'm no Liz Socket, and much respect to Liz, who's make wonderful movies of this, because I'm such, I'm not going to use the English expression for it, which is very evocative and brusque, but I'm not a very good microscopist. How's that? And so, because they're so tiny and my eyes are so bad, but literally, and I've seen it when they hit a prey cell, like it's knocked way over there. So do you have the same kind of kinetics that you're seeing with the Curli containing prey cells?
I think we need to do more microscopy. We have not really done too much of that. We kind of did, I would say, more time points and less live cell, because we were kind of encountering some of these technical issues of tracking everything.
Sure.
So now that, we're up and running in this lab, that is one of the main things I'm working on, is trying to develop some microscopy tools for Dello so that we can ask, I would say, kind of the next level of questions we need to ask for this model.
There's a wonderful paper from quite a while ago. It's highly mathematical, so I don't understand it, and my wife, who's a mathematician, does.
Yeah.
But it's about decoys in predation.
Okay.
And if you don't have that paper, shoot me an email to remind me, and I'll send it to you so you don't, so you can actually have a look at it. And I don't know if it's relevant, but you may be thinking about this. What's going on. It's just so fascinating, and I don't mean to sit there and quiz you like it's a seminar, but
Oh, no, you're good. This is my favorite thing to talk
I
about.
love this. And Mixo, as I say, is wonderful. And you're aware that some bacilli become resistant to Mixo because they build the castles. Do you know that one?
Yes, I love that one.
And again, for listeners and viewers, why don't tigers rule the world? Because the prey of tigers learn or evolve mechanisms to evade that predation. And the same thing is true at the microscopic level. And that's what we're talking about for this whole seminar.
Yes, definitely. Finding ways to fight back. Everyone is just trying to survive, especially I think at the microbial level where it's just your one cell that you're left with. You're always just fighting for that chance to survive, trying to find those new tools.
Hannah, one thing that occurs to me is whether or not the focus is on individual prey cells being attacked by an individual predator cell. So, for example, when Vibrio goes after E. coli, is it doing it in just one particular thing? Or what about collections of prey organisms like you might find in a biofilm? Is there an overall protectiveness if some members have curli and some members do not?
Absolutely. We looked at this a little bit, and we were able to look at this more in a liquid culture, so not a true biofilm, but we were able to mix together curli producing and non-producing cells in that liquid culture, and specifically saw that Delo could kill off the non-producers, but the producers were protected. So it's not quite like they can encapsulate those free ones, or it's not really a community resource, at least in the liquid culture. I would say we haven't been able to really get the setup to interrogate this as well in a biofilm, especially with the idea that, biofilm's going to be layered and would each of those layers kind of have different susceptibility. We're still trying to work that out, but what we are seeing is that some E. coli do seem to upregulate curli production in response to Delo. And so we do think there might be able to be also a community level that we're still trying to work out. So far, it's been single cell one-on-one, but we're hoping to better also understand this community dynamics. I definitely think that's going to come into play as well.
There is an ancient paper called A Bacterial Scream. Have you seen it? So it's early work with gene arrays. And the question was, when they're exposed to Vibrio, what genes are turned up and turned down? You should have a look at it. It's called like a bacterial scream.
Oh, fun.
If you can't find it, holler, because I got all these.
Oh, fun. Okay, you'll have to look at that.
And I love what you're saying there. The person that we had on last week was showing how different members of a biofilm compete with one another in real time. Now, you could do the equivalent with live dead stains in a biofilm of E. coli, I'm sure, because the curli strains do make biofilms.
They require both curli and cellulose to make biofilm, so it is two components, but we can use the genetics and tools to kind of play around with all of that.
And how cool to have a patchwork, a mixed biofilm, and see whether some areas are, this is lovely work. Too bad I'm not much younger.
Yeah, come do a postdoc with us. It'll be great.
Well, it's not called at my age. It's what we call not a postdoc.
Postdoc, there we go.
It is so fascinating. And you have the advantage, and I need a German word for happy for you and jealous, because there has to be a German word like that.
I'm sure there is, yes.
Yeah, because it's quite a language, isn't it? And because you can do things that I always dreamed of doing. And so I so look forward to following up on this stuff and seeing where you go with it.
Oh, thank you. Yeah, we're really excited, and we can't wait to, get things up and running more and get a bunch of students in the lab so that we can start kind of cranking out some cool, exciting science.
I couldn't agree more. Tell me about some other, I feel like I've talked about things that interest me more than anything else, here's a hint, which is one reason I do this podcast is so I can do that. Point of being disclosed. But I want you to have an opportunity to talk about things that interest you that might not be involved with Dello or Mixo.
Absolutely, yeah. So the other half of the lab is really excited about the ways bacteria defend themselves from phages, their viruses. Here we have a really kind of a narrowed, focused in on these systems that have homology with components of the human innate immune system. And so we actually found that bacteria perform all of this biochemistry we thought was kind of unique to eukaryotic cells. So in my postdoc, I found bacteria are performing something similar to ubiquitination to regulate their immune systems. And we had no idea bacteria could even do this. And so we're better trying to understand the biochemistry behind those systems. And then just in general, we're fascinated by the idea that there seems to be kind of the best way to do an immune system. I wrote a review that we can link to that's really comparing immune signaling across life. And we just keep seeing these same trends of what seems to be the way these pathways kind of come together and the most efficient way to signal that there is a pathogen around. As we're kind of trying to define what are those key steps with the hopes then that people who do more translational medicine could come in and kind of help find intervening steps at those things to help boost human immune signaling for whatever disease. Along with that kind of idea and sticking with the Dello aspect, one thing we are just beyond excited to try to understand is now how can Dello overcome curli? Everyone's fighting back. We were talking just now about how we're all trying to live. So we know that there's now going to be predatory bacteria out there that have evolved ways to overcome these amyloid proteins. And what cool biochemistry and biology can we uncover by learning just from the source? How do you manipulate amyloid proteins? These are such a massive biological problem. And why don't we just let the experts tell us how to do this? And so we're kind of really digging into the functional amyloid biology, both from the host and the pathogen side, to define kind of how we can manipulate these proteins. And so that's something I'm extremely excited about, and we're trying to get up and running in the lab. And here, this is one of my favorite things we're doing is we're working with the undergrads at the university, and they're just going out in the dirt, and they're trying to isolate predatory bacteria directly on amyloid producers to find ways that they can overcome. So this has become this really exciting kind of educational outlet I get to do as well, to involve a lot of undergrads in the lab and trying to explore new aspects of science. And it's been so much fun.
You call it hunting the hunters, right?
Oh, I'm going to now. Yeah, I had not called it that. I love that.
It's true. And it's, if you think about it, look at the CPhages program. That's a wonderful cure system where students are asked to go isolate bacteriophages. This is no different. And it might have indeed medical relevance because many people have talked about using predatory bacteria. Please don't see me get too enthusiastic as, quote, living antibiotics. I have many thoughts about this. There have been some successful chases, but I think we need to understand how it's doing things. And that's no different than when you get a new antibiotic. You have to know how it's working before you can think about using it clinically.
Absolutely. Yeah, that's an avenue we're kind of really entrenched in, both phage therapy and then the idea of using predatory bacteria as a therapy. I think, like you're saying, we really need to better understand the biology of how these interactions are happening. And so that's where we're really excited to hopefully make some contributions. Can we better define exactly how all these organisms come together and work so that then we could exploit it for some more medical relevance?
Yeah. And sometimes people will say things to me like, what are the applications of what you do? And I try to be very careful about that because, as I've mentioned several times, I'm ancient, and I remember when we would talk about bacterial gene regulation on grant proposals, and we'd say, well, this might be relevant to how cancer happens, which is not true. But that's what you wanted to say at the time. So in a similar way, looking at this could eventually have therapeutic approaches, and people have tried to do that. And I think it will really help a lot because I think pure research brings us a lot of interesting things for applied, but you still have to support the pure research.
Absolutely.
And this is a great way to think of it.
Wonderful. Yeah, I love that. And also I feel like we've just kind of seen mostly through the study with phage that, bacteria just seem to be a gold mine for biotechnological tools. And so
Yep.
I love studying microbial warfare because you continuously see how our basic research is able to be translated into helpful tools, CRISPR being, the mighty one that
Hmm.
we can refer to. Um, and so it's just always exciting to kind of envision what could the cool tool be that we'll uncover next. And while we don't develop the tools ourselves, it's always very exciting to kind of think about that potential.
Well, I get called a microbial centrist quite a bit, and I am, and that's because they were first evolved and they'll be last extinct, and the approaches that evolution gave them, the hand of Darwin's on all that lives, right? And so if you think about it, look at the things that the microbial world has given us, and yet we don't talk about it enough in classes. Now, let me explain what I mean by that. We don't have a microbially centric introductory biology course, and we should. Now, someone says to me, anyone could say that about whatever system they like, and I could say, embrace the power of and, please. But I would also say that it's at the it is the basis of the very biosphere. And that's something that I've always tried to hammer on the students. And I'm glad to see that I have a fellow microbially enthusiast I'm speaking to today.
Absolutely. Yes. I was just teaching a graduate level class and we were talking about eukaryotic biology and I just kept reminding them that,
Yep.
it's just how the bacteria do it, but more complicated. But bacteria do it the same way. It's just simpler and more approachable, I would say.
I was talking about sigma factors in my microbiology class, and a student
Yeah.
raised his hand and says, well, those are like transcription factors in eukaryotes. I said, no, quite the reverse,
hmm
right?
That's exactly what we were talking about in class. Funny enough, I
Yeah.
was comparing transcription factors to sigma factors. So yeah.
Yeah. And I'm not being negative about eukaryotes. There are three of them that I'm very fond of, right? My wife and
Yeah.
my two sons, but and all my students, and I'll include you in that area.
Oh, wonderful.
So what I'm trying to say is embrace the power of and. I mean, and you've heard me. If you've studied me at all, Lord help you if you have. But I talk about the problem we have is eukaryocentrism in the curriculum, and
hmm
then I would add to that oxycentrism. And then for my smug microbiologist friends, I would add coli-centrism to that.
Yeah.
Because let's face it, nature has so much to teach us.
Yes, absolutely. That is kind of the model in the lab is look for something weird in a weird bug. That's what we want to study.
I
So we
couldn't
can,
agree more. And there's that famous Isaac Asimov, the late science fiction writer, had this
Mhm.
wonderful quote, and I've said it in several podcasts, and I'm going to say it here, that, discovery doesn't happen, and this is vintage Asimov, when some damp Greek emerges from his bathtub shouting Eureka. It happens when someone looks down at the lab bench and says, that's weird. And that's 100% true.
So true. Couldn't agree more. I love that.
So as we start to think about winding down a little bit, would you care to either, it's up to you, talk about your path to the microbial sciences, because many people think there's only one, please, or if there are several take-home lessons that you would like listeners and viewers to remember?
Ooh, let's do the path because I feel like, I
Okay.
found microbe. Yeah, I love microbes. Let's talk about that. Okay, so how did I find microbiology? It was late in the game, I would say educationally. I really only kind of figured out my obsession with microbes in my junior year as an undergrad.
Mm-hmm.
Like many of us, I was pretty adamant I was going to be a medical doctor, and that was kind of the focus. I was very eukaryotic centric, but it wasn't clicking. I didn't feel that like, wow, I can't wait to go to class. I can't wait to understand things. So I started just kind of meeting with a lot of different professors and sitting in their lab, trying to just get lab experience. I knew I liked to do hands-on things. And I was sitting in Dr. Vishwanathan's office at the University of Arizona, and he explained to me how E. coli come into our gut. They can bind to our cells, use their secretion system to inject proteins into our cells, and using two proteins can completely rewire whether or not that cell lives or dies. And I had never heard anything so cool that this tiny, tiny little thing that I had never thought about in my life could completely rewire one of my cells. It just was this aha moment of like, I need to be in this space in some way. I need to be in microbiology. And so he was wonderful. I was able to work in his lab for a year and a half and really start exploring the wonders that are microbes. And I just never left. I would say the other thing that really drew me in is I read the book Microbe Hunters. It was my postdoc advisor in that lab. He handed it to me and said, "You seem nerdy. You should read this." And I ate it up in like 2 days and was completely obsessed with these tiny little creatures that we really just don't understand. I think the appeal for me was, so much of our education, we're taught like we understand everything and we already have the solutions. And when I got to microbiology, it just seemed like, nope, we're full of open questions and you get to ask the weirdest, most creative question you can come up with. You're not locked in kind of in the types of questions you can ask. And so that's really just kind of what's kept me excited is something my previous mentor and I always say is, if you can imagine it, there's already a bacteria out there doing it and doing it better than you imagined you could do it. And so you just get to remain childish and creative, and you just get to play make-believe a lot. And it has just kept me coming back to work every single day because it's just so much fun to think about the weird things that bacteria could do and then try to figure out how on earth you could prove it.
I mean, even at my level in the trophic web of science, I tell students there's no substitute when you found something out that's always been there. Let me be clear, but you're the first person to have seen it.
Yep.
I didn't say it was super important, although
but it was
sometimes,
yours.
But it's yours. You did that. You found that. It's like Leeuwenhoek felt the first time he looked through that microscope. And it's transformative. And there's nothing that brings more people to the one true microbial faith than that level of discovery.
Absolutely.
And I think you, like me, are a microbial evangelist.
Yes, I walk around my department is not a microbiology department, but I'm going to convert all of them into being microbial enthusiasts by the time I'm out of here. So
it has to be that way because that's the center of everything. It doesn't take anything away from eukaryotes. I love tardigrades. That's a eukaryote. It's even multicellular.
Yeah.
Yeah. I've got one right here. So, and you will receive one shortly too. Did you notice it has a hat and glasses?
Oh my gosh, so cute.
We call those tardidocs after me. Yeah. Okay. So in any event, what I would like to as we start to finish up point out is that you have such enthusiasm and you've done such important work that's been at the center of things for so long. Incidentally, I was not asked to review your paper. I usually get called on every Delavibrio paper in the world, and that's because I'm always very polite, which I hope everyone was at Nature. Reviewer 2 is the one you worry about. I get that, right?
Yep.
I love explaining that to students, by the way, but I think it has been a wonderful session with you today. I can't wait to see what you do next. I have so many things to tell you via email or something. Yes. And do you have a final piece of advice for listeners and viewers?
Ooh, my final piece of advice is, pursue what makes you excited. It makes life worth it. If you're happy about what you're doing, you'll find a way to do it.
Well, I really appreciate having you here. I am so proud of your new position at the University of Michigan. And Ann Arbor is a lovely town, by the way.
Absolutely beautiful.
I need to get back out there. I have many friends there, as I've mentioned.
Yeah.
So I want to wish you and your lab group, and incidentally, how many people do you have in your lab at present?
We have 2 full-time members now.
So I'm going to send you a number of stickers. Okay.
Okay.
That's what
excited.
I do. Okay.
Well,
anyway,
in our lab.
Thank you so much for your time, and I hope to chat with you soon.
Yes, thank you so much. This has been so much fun. I really appreciate it.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with beyond epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Hannah Ledvina can be found in the Molecular, Cellular, and Developmental Biology Department at the University of Michigan in Ann Arbor, Michigan. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today, and see you next time on Matters Microbial.